Okay, we should get started so we can finish at some point. Just in case anyone is interested, you can catch it at 1 o'clock ferry home. So if you also want to catch the 1 o'clock ferry back, there should be a taxi showing up around the house. Yeah. So this session we're going to start talking more about reproducibility. We can measure these things, how we can help people. And then we're going to start with Gary telling us about functional curation, the cardiac electrophysiology. Thank you.
Okay, so I'll better have just a quick show of hands. How many people saw the demo first time around last week? Well, not that many. Okay, I'll get on with it then. So just a quick outline. I'll give you the motivation for this work. What the web lab lets us do. Give you a quick then talk about future stuff. So this is my vision of the future. Is that biological knowledge is represented as a quantitative mathematical model. So if you want to describe for someone how the channel works, you give them the mathematical model of it rather than a few summary graphs. And so experimentalists need to be able to, in that kind of way of thinking, do the right experiments to fit the model. We're not a million miles off this in ion channel modeling, so I don't think we need to do too much before that could be a reality. And when something new is discovered, so maybe they work out that something gets phosphorylated or whatever, then the protocols that should be performed would be the protocols that describe the channel, then we phosphorylate it, then we run them again, kind of thing. And so all this sort of starts to fall into a framework of how should we make a model. It's a bit of an art at the moment. It should be a science. So I've written a little story about what a vision of the future looks like. If you have a look at my blog thing, if you're particularly bored during the rest of the talk. So we're all familiar with the model databases, biomodels and Physio repository, with CellML and HTML describing protocols. Describing the models. But what these repositories typically look like is like this. So you've got lots of different models that you can, if you're lucky, just plug into a simulator. But inside each of these models, typically there's just sort of one hard-coded protocol. So for the electrophysiology stuff, that's typically 1 Hz steady pacing will be hard-coded into the model. And to do anything else with it, the first thing you have to do is kind of mess around with the generated code to unpick that and do something else. So what the web lab tries to do is separate out this idea of protocol, or what you're going to do with the model, from the model, leaving the model as just a kind of hypothesis about how the biological system works, and then the protocol for how we intervene with that. And we call it a protocol because that's kind of what they call it in a web lab. They write down a protocol for how we do an experiment. So the link between these is these annotations that are just the same as the ones I showed you from the Chase CellML files. And And then a simulator will pull in a model element protocol. And perhaps the protocols can have some library giving them common components. So the language that we've come up with for the protocols is sort of CellML based. So it will actually accept CellML protocols, but it also uses some of our proposed extensions to that. We have a text-like simplified view of the XML, a bit like OpenCore. And you define the interface that you're going to use to the model, so it might be as simple as I'm going to specify the voltage and I want back sodium current. And then the interface is sort of built on the fly, a bit like SemGen, I think. So you can inherit bits of protocol from other protocol files or libraries, just like CellML 1.1 does for models. For example, if you want to do an action potential duration calculation, which ends up being quite a fiddly operation, you don't have to define that every time, so you just pull that in. You can perform nested simulations, so loop over things, changing parameters. So George might find this a neater way than putting in that ODE to do it. And you can even call whole other protocols from inside loops. So this will all be a bit clearer when I give you an example. So you've got the kind of interface, what you're going to do, and then post-processing is specified as operations on n-dimensional arrays. So once you've come out of loops of nested simulations, you end up with these n-dimensional arrays. So it could be voltage against time, but then against some parameter you're varying. And so those can be a little bit tricky to think about, and you're doing all these windows and slices and reductions and things. And then we have plotting with some simple options. And all this is kind of what we call that the functional curation sort of language underneath, and then the web lab is a web front end to it. Some of the nice features, units conversions, we've talked about that earlier. Once you've specified the inputs and the outputs, the simulator works out which equations from the model it actually needs. So if you clamp the concentrations and you're just doing a voltage clamp and you want the sodium current, it'll throw away 99% of the model. You can replace bits, so if you have got a stimulus current in there, maybe you want to replace that with a different stimulus function. We use Plot, which I think it was Google, but might be independent now, and then or some other graphics libraries for the plotting. If I don't show you, remind me to show you how we can drag metadata onto the CellML files from inside the web lab. And when you've finished doing combinations of models and protocols, there's a combine archive is made at the end of that. We heard that yesterday. So now we'll try a demo and basically make it work. There we go. How about right. Okay. So here we have a big grid, which can I make it bigger? So we've got a great big grid with a list of CellML models down the side, and a list of some protocols that we've come up with across the top. And then this is color coded, so the ones that are white, no one's running it. Someone's actually trying that out and putting a new model there. They've just run one experiment on for now. The green means that the protocol worked and ran to the end. So most of it's green, which is nice. The grey means it's not applicable, so the protocol maybe asks for something like a fast transient outward current, and a lot of these models don't have a fast transient outward current, so you can't do them, so they go grey. Orange ones mostly work, so this should be something like asking for an action potential when there's a flat line. It might say no, you can't do that, so you can get an orange, but mostly worked, and red when something actually went wrong with the solving. So, and this is actually that fancy Rudy model that someone asked why is there an outlier in quite often breaks CV over either. And so I'll just select, you can select a few experiments to look at. So you can get a little sub grid up. So if I just select a few of the human models for us to cover. Yeah. You get a submatrix, hopefully. Okay. Another quite nice thing is you get a unique web address for all these things. So you can always get back to a particular view that you're on. So I'll just copy that address in case we get a bit lost. So now if we click on comparison, better models. I've done a bit wrong. I've selected too many things. We'll just do it. Okay. So I selected 1 Hertz pacing there. And now it's going to compare for us all these compare selected experiments. There's a kind of default plot specified at the end of the protocol file. And what they should do, when we've communicated with Oxford and pulled all the data back, is plot you the 1 Hertz action potentials for all those different models. So you can, this is actually the first time I'm aware of that you've just been able to go and have a look at what an action potential from a CellML model looks like, which is quite handy. So, so that will be comparing lots of different models with the same protocol. We might want to do something a bit different and say, if I compare an O'Hara model at different pacing rates, we should be able to just pull up different action potentials there. So we just plot 2, so in case there's any alternate or anything, but then you can have a look and compare the different protocols. So you get the full flexibility there. We have a look at something a bit more complicated, like blocking the channel. So here we run the steady action potentials, but that's also nested in a protocol where we increase the percentage of IKR or third block. Then you can see that a lot of models will suggest 20% prolongation of the action potential. Some will get to pre-8, there we'll get to 80%, but then stop working. O'Hara will get to 90%, so we could dig down and try and work out what's going on there.
Here, can I ask? So that what's being plotted out is also nested in that protocol. Is that correct? Or
it's so the protocols, so the plot's specified at the bottom of the protocol file.
Yeah.
What's being plotted out, and then the web lab just, if it sees the same plot, will overlay them if it's got the same name, basically. They'll get overlaid in the web interface.
so you can have a similar upper protocol and maybe plot different things.
Yeah.
It'll be a separate protocol, right? If you apply it.
It'll be the same protocol because you have different plots. So down the bottom here in this section, this is the sort of main summary graph. But if we click on this one, we can go and look at the individual action potentials that we're beneath. So hopefully, I think the delay from Oxford to New Zealand.
So Gary, is it running simulations?
No, they're sort of run beforehand and then they're stored in a database on the web server. So, so the ones, the little green square or the little squares mean a database entry for that experiment. So here we can see why we failed to get an APD for the biggest block, because it turned out it was 100% block gives you this, that should come up on top. Yeah, you get the idea. Then you can zoom and select bits, and put the usual bits and bobs. And you can save any file. These are slightly different visualization libraries. You can use whatever you want once it's in there. Display the contents, download stuff, and then the archive at the bottom is the combine archive. Oops. So that's most of all I want to show you. You can just go back to that comparison of them all. And underneath this, we store all the different versions. So if we click on a model, you can see the different versions of it. So you can see I've edited this a little bit. We can compare the versions either with just a straight diff or with Martin, who's on the call. He's got this Bivs by VS, which is how do we say it? Beavis. And you can see what I've changed. So all that's changed is I've put in some of those metadata tags on the different versions of the model. This is quite nice. It shows you the maths in a nice way and things like that. I suppose I should show you quickly what an actual protocol file looks like. So something like an INA IV curve. So if I click on this, I can actually have a look at it. This is what the text view actually looks like. All that just generates the markdown documentation. Some little bits and bobs to make this work. These are all the inputs that go into the protocol. So these are like protocol parameters. So you can change any of these when you call it. So we've got a holding potential, amount of time to get to a steady state, and then all the test potentials for peak, inactivation, activation for protocols. I have to define some of those units. So this is something we could pull in again, but to make it work with all the different models, we've got to define quite a few different units there, just as you would in CellML, really. And then here we have all the different interfaces of the model. So in this one, we actually clamp all the concentrations. So we say that we want to know all the model's concentrations. And we're going to output sodium, voltage, time. So we could have optional things actually, which is quite nice to work with old models. So we might want to make sure temperature has a default, but it's optional. The reversal potential, so the really old ones don't have concentrations, they're reversal potentials. So you can say if it doesn't have a concentration and it does have a reversal potential, we'll fix that. Clamp various things. So this is quite a complicated model file in the end. More units, conversions from membrane codes. And then this is the actual tasks. So task 1, run the simulation until steady state's reached. So we just say we're going to range over time, and modifiers happen during this range. We just say at the start, set the membrane voltage equal to the holding potential, and save the state as the holding state. And then once we've done that, we've worked out what the kind of start point is. Then we can loop over different test potentials. So each loop reset the holding state, and then set to the holding potential. And then we loop over getting peaks out of voltage. So that's actually that's the peak current. Voltage protocol is that easy to define. Then we try to do inactivation and activation underneath, but that's getting a bit complicated. And then these are the n-dimensional array post-processing, which is a bit difficult to work with. We try to go with a kind of Python style, first index, last index, remove dimensions, take maximums, that kind of thing. It's easiest to copy off one that's already done. And then you define the outputs you want to go to file, and then you can define the plots in terms of all the outputs. So when you actually so it sort of follows the CellML way of doing things to a large extent. And the results of that protocol, you compare all the different models, would be this should be a sodium IV curve. I've probably been talking for too long now.
Question.
Yeah.
So kind of back to the beginning setup where you had a whole list of models, and you pointed out that typically the repositories contain these models with protocol. So how much work was it to take that out? And
Well,
what work did you have to?
we haven't, in a way, we haven't taken it out. We just make sure that, so it's still in there, but we'll overwrite it. So if we have a protocol that depends on the stimulus current, we overwrite it. So we haven't actually had to manually
So
unpick.
you didn't change its original?
CellML's original with just a metadata tag.
And it doesn't.
So if the protocol includes stimulus current, and we want it to, then we just make sure it has the properties we want in terms of the period or whatever.
And wouldn't you get one also? Have you turned that one off?
Well, it knows that, because it's got the same tag, it knows that that's the one it needs to be. We overwrite the equation.
So when you generate the code from the CellML model, you're then working with the
Not so.
Yeah. Yeah. So we get a bewildering variety of sodium IV curves. We can start to compare with experiment sets. Okay, I'll go back to the. Yep. So in the future,
I've only just done 20 minutes, that's quite
in the future, I'd like to join this in. So experimental data is attached to these protocol definitions. So that these want to be as much like a wet lab experiment description as we can make them. And then it becomes really powerful because then we can start to compare with the real data. We can start to do things like actually fit to it. So what the short-term task, we want some kind of an editor for making these protocol files. At the moment, you have to do it and see if it works. And the debugging is a bit difficult because you get slightly obscure error messages if it's not. Better documentation. I've got these off John Lucas on the lab to say that. More flexibility in the protocol language, but we're a little bit afraid of getting too far ahead of CellML and creating too much of a gulf, so we'd like to bring it along with us. John's an editor on CellML, so that's how we're tackling that. Rather than an Oxford server, this could move on to a cloud like DMR. The ontology, it'd be nice to have a bit more structure in it, and that would simplify things because we could say commands like all the cytosolic concentrations, and we wouldn't have to name them all and go through it all, or something like plot the sum of the transmembrane currents or potassium currents. And so you can see we wouldn't need a very complicated ontology to start to capture those kind of things. So we'd like to get that in sooner rather than later. And we'd like to start being able to plot against experiments. It's worth mentioning there's this minimum information about a cardiac electrophysiology experiment, which is a lot of labs signed up to, but not many have really got into putting entries in the database when they do a real wet lab experiment. And ideally, you want this to collide with our protocol definition so that we could actually start to automatically get machine-readable protocols from the experimentalists without much of intervention. And so this explicit link to experimental data will start to allow us to fit parameters automatically automatically modify models, improve them, compare them. So things I would like a bit of input in. Adding our metadata tags to the repository, we can probably talk about that in a bit. Getting to one reference version of CellML. I don't know if there's a nice easy web service or something so you can just say to PMR, what are all the models? Give me the one with this name. Perhaps we could use some of these to enrich the curation of the electrophysiology models in PMR, so maybe click through to explore some of this. And anything you'd like me to report back to John on different standards for data and protocols and biosignals and stuff. Okay. We'll have a workshop on the web lab in Oxford, 10th to 11th of September. So that should cover all aspects of using it, uploading your own models and protocols, or using the underlying software in a command line way. And that'll be an opportunity to steer things and get involved in future developments. And as always, most of this work is John's. Martin did the web lab prototype front end to all this. That's it.
So, I use software probably from that. So what has to happen now for setting up to adopt extensions? Is it that we've got to implement it in another code?
Yeah, so I think it would be that our proposals would.
Could be that.
Yeah, could be. I guess the most handy thing for the CellML community would be if it gets into Frank's CellML API. And so we have a couple of codes that can understand that. And they've already, since we developed it, things like nested protocols have made it into CellML, so there's not much. With the further behind as time goes on, they've moved together quite nicely. I think it's quite possible to have it stay a step.
Have you guys done anything with anything other than electrophysiology?
No, electrophysiology has been tested mainly because it's unusually consistent between models. So most of them have got sodium current, for example, so it starts to make some sense to compare. I guess it's my main interest as well, so I'm pushing it along those lines.
So I just want you to find me doing it right, because I didn't understand your answer about how we override protocols. So do it all, but find it.
Do you have like top rankings of a model that has been downloaded? Like data?
No, so this isn't the main repository, this is just our version. One thing that people quite often think that we've done is put green things by good models and red things by bad models, and we definitely don't want to put my head on that block. So these are just models where it ran or it didn't run. There's no judgment.
Okay, moving on. And Dan's going to tell us about virtual reference environments, reproducible research.
Okay, we have time. All right, 15 minutes. Firstly, thank you to Andre and to the organisers for giving me a chance to speak. Thanks to all of you for listening. I think, looking back over the stuff we've talked about over the last day and a bit, what I'm talking about here is, I guess, kind of supporting infrastructure, which will kind of slot in underneath a lot of the initiatives and the software tools that we've talked about. I think that's really neat, and I'm really excited about the opportunities for some of this work, as you'll see, both with the SimGen stuff and the work that Gareth did. So I'll get started, I guess, first by talking about the word reproducible research. And reproducible research is kind of an overloaded term, as I guess a lot of you know. And as a result, many people have produced different, more specific terms like recomputation or replication and so on. And this is understandable because there's lots of ways you could think of research as being reproducible. And the way that I first thought of it is that there's kind of two different conceptions of reproducible research. And I call them the recipe and the snapshot. And as you'll see, both of these I think are important. But often when people talk about reproducible research, they're actually talking about one or the other. And part of my thesis here is that they're related. When I say a recipe, it's like an actual recipe. That is, it is a minimal list of instructions in some sort of language, preferably human readable, which will allow you to combine a bunch of ingredients to get a particular result, to do a particular computation in our space. If you're feeling really good, then your recipe will possibly involve some kind of literate programming thing. If you're in the R space, you might have heard of Sweave or knitr. And Python is IPython notebooks. And what they do is they allow you to generate both your paper artifact, your published paper, and the figures thereof, and also your code and the computation that you're doing to get the result. And they unify it. And this is really neat. This is the recipe aspect. The other aspect, though, is what I call the snapshot. And this is much more the replication or recomputation or archival version of the result. That is capturing, and this is what I'm going to show you in a sec, capturing a minimal software stack required to repeat a particular result. Now, we've written a couple of papers about this, and although this is going to be quite a software engineering focused talk, there's a lot of quite interesting philosophical, I guess, and structural and policy decisions about what we should actually replicate, what we should capture, and why. You know, the licensing implications and privacy implications and all kinds of stuff like that. So lots of actually quite philosophical stuff to talk about, as well as the computer program. The other thing that we need to also think about, as well as replication and reproduction and all that, is deployment. We must always also consider the idea that if we're reproducing these things, we want to be able to deploy them or distribute them in the most appropriate way for a particular situation. Increasingly now, you know, as you all know, we might want to run code on our local machines. We might run it in a centrally managed computing facility in our institute. We might run it in a cloud somewhere. But ideally, what we want is to have confidence that we're doing the same thing in each one of those situations. You know? And we don't always have complete control over those environments, but we want some confidence that if we do something in the cloud, we're going to get the same result as we did on our own machine. Okay, so this all started actually when we were publishing a bunch of papers last year and wondering about how to distribute our code, but also how to motivate people to actually download our damn code and use it. Because there's a reason, downloading someone else's code and using it is a lot of work. And you need to really incentivize people to do that. So one of the things that we came up with, and it seems very obvious in retrospect, was look, we can make, for results which are produced entirely using open source software, we can make a little reproducible, bootable environment which has a minimal installation of some OS and just the things needed to reproduce a particular result, a figure or a computation or whatever. And that ended up, we did that in a few papers, and that ended up becoming an opinion piece, this one in reference to bioinformatics. And this piece is actually deliberately a bit controversial. I like writing things which are a bit controversial, because in here, we're kind of saying, hey, if you can get a result in computational biology or bioengineering, then you can create a minimal environment which will reproduce all or part of that result. Now, I should, because this will come up in the discussion, I should qualify that by saying subject to licensing requirements, privacy requirements, and practicability of the availability of your data and so forth. But there are various mitigations to those things. So what I'm going to do is show you one of these, what I call a virtual reference environment. And it's going to be a specific example, which is based on this paper, which is the one that one of the ones that Edmund discussed yesterday. This is the bond graph representation of the lambda-fin Kushmeric glycogenolysis model. Thank you. And with a little luck, I can. That was a slide in case we couldn't get it working. We don't need that slide. Okay, so what is this thing? I will boot it up and carry on talking. What it is a little potted virtual environment. In this case, it runs in
Oracle's VirtualBox, which is a pretty common Windows and Linux-based virtual machine engine, but you can also produce ones for Zen or VMware or various other virtualization solutions if you like it. A big part of what we're trying to do here is to be technology agnostic as much as possible, because what we've found in Edmund's group is that there are people who do most of their work in Python or in MATLAB, but there are other people who have more of biostatistics, bioinformatics focus. They work exclusively in R. If you do work with proteomics or mass spec, there's actually quite a lot of tools in that space which are Windows only. So you end up having to do a lot of work in Windows. So here we go. I just minimize that and bring that up so we can see it a little bit. So what you saw just then was this tiny little Linux environment, which is about a 700 meg ISO or so. This is a deliberately chosen small example, but this is an ISO, a read-only file, and there's a reason for that, which I will talk about later on. So what we can do with this environment, and it's based on a minimal Linux distribution, and as you can see, it has a fairly minimalistic Windows XP-like GUI. You don't need to have a window manager, of course, but what we found is that for accessibility reasons, if you have a window manager that looks a bit like Windows XP, look, there's a button you can click down the bottom. It's got some things and a web browser and stuff. So if you have something that looks a bit like Windows XP, it's a lot more accessible for people who aren't necessarily computationally focused. And this is quite an inclusive activity, right? We want people to be involved in this regardless of whether they are computationally focused or not. So we can run the script, and bang, all kinds of science will happen. So what's going on here, just for the sake of argument, is that we are executing the code that Peter and Joe and Edmund created to extract parameters from this language encryption model. It's on the local environment, it's not going on the web. And it will generate a series of figures and a PDF at the end, and it takes about 3 or 4 minutes. So while this is running, one of the things that I wanted to point out is that this is very much the snapshot example of the rest of the labeling in. This is an ISO, what used to be called a live CD, and as you can see, you stick it in the drive, metaphorically speaking, and it boots. The reason for that is that this is a read-only archival kind of thing. You can check something, and that means that you can warrant if you're, and we're moving away a little bit from the research science angle, but for a policy or legislative or maybe a clinical situation, you can warrant that a particular environment will give a particular result on a certain set of data, given that checksum. And that's quite important. The reason for that is, as you know, tomorrow dependencies might change, or a particular version of something may or may not become available. A certain operating system will become end of life, all of those kinds of things. This way, we can keep this environment, which produces a particular result, in perpetuity. George, I looked during the break, and there is a PDP-11 emulator, software emulator. So we could, if you can remember what you did, or if you have printouts of your code, we could, in theory, replicate that in perpetuity. I'm sure it's worth keeping. And I mean, in all seriousness, I would struggle to compile the code that I wrote 10 years ago. I had some of it, and I wouldn't even bother trying to compile it. And yet, I have a Commodore 64 emulator on this computer, on which I play Commodore 64 games, and they are 30 years old. And this is really important, because as I was saying to Mike during the break, it's quite possible that 10 or 15 years from now, given the avalanche of data we're getting in a variety of places, the results that we are getting now are going to be just as important as the data that we are using from the 1970s. Right now, we are using, in many cases, data that dates back, or models that date back to the 1970s, and we're having to re-implement them. As we get more and more complex, that's going to get more and more difficult. So having a snapshot, an archival version, has got some value. And the key point here is being able to do it easily. So we can spin one of these things up in a matter of days, given a particular set of code, producing a particular result. And here, although the res, sorry, I'm running away, res is a bit unpleasant. This is popping a PDF. Again, we don't have to do this, but it looks nice. And you'll see it is auto-generating the equations that I think it has taught. And it's generating the actual figures, the bond graphs, and so on and so forth. And these are the artifacts, the work products that end up in the paper. And really not super difficult. So, okay. Has anybody got any questions about that just before I move on? Cool.
Do you provide the source code for the model?
Yes, I mean, this is an adjunct to the normal release of code on GitHub or SourceForge or something like that. As I say, two reasons for doing it, but two key reasons for me, I think, are one, the archive in perpetuity, and two, motivating people to actually go and code. Because, you know.
And kind of a quick question. Would someone using the virtual environment be able to modify your model and then rerun the replication?
They would, yes, although this is, because this is a live CD, you would need to copy things on, so every time you reboot it, it's completely fresh. We deliberately, I mean, it has a networking stack, you can get out to the internet, it has a browser, etc., etc. So this is, if you like, the entry level to try and do something. It also has a debugging or a troubleshooting facility value in the sense that if you have a working example of some code, it's a heck of a lot easier to get it going on whatever environment you happen to be on. Because you can examine the differences in a principle level. Okay, so back to Bill. For a second. Cool, okay. So we have produced a number of these. The other thing that I would point out here is that for the sake of accessibility, what we are doing is printing out some simple instructions and also timestamping it. So you remember I talked about this conception of the recipe and the snapshot. Ideally, what you're doing is you are linking the two because you use the recipe to generate the snapshot. And there is a hard link, if you like, between the two because the snapshot says this was generated using this version of the recipe at this time. And as you can see down the bottom, we're saying this is the last version checked in any code for this. This is when it was, and this is when the environment was made. So, how does all this work? So I'm going to delve into a little bit of the sort of software engineering detail, and I'll talk you through this using a bit. The initial part, I guess, is building, using this configuration out of the repository, building a base image, and just focus on this part here for the moment. To focus on this part for the moment, this is the virtual machine aspect that we just looked at. What we're doing here is setting up from, in this case, the Ubuntu install ISO, it's Ubuntu 14.04, 33-bit version, installing all of the core things that every environment will need. So a compiler, Git, SVN, WT, those kinds of things. I'll go on and talk about different ways of deploying things in a minute. Following down through here, we have set tests which ensure consistent behavior. These are shell level tests, where we're trying to make sure that things install properly and that the environment behaves at a very basic level the way we want it to. Those are automated. Then we have project-specific configuration. This includes not only the code and the data for a particular project, a particular artifact, but also the tools that are required specifically to get that result. So Python, if you're using Python, R, if you're using R, whatever. Then we have another set of automated tests, which are project-specific. Generally, these will involve something to do with the actual science, the actual result that you're expecting. And finally, we can distribute these in different ways. If we go back up here for a moment, see that I am talking about not only a virtual machine image, but also a couple of other ways of making things. If anyone's familiar with the container tool Docker, this is an implementation of what's called Linux containers. It's really intended much more for deploying server applications, and it's kind of an intermediate step between running a virtual machine and running something natively on your underlying command. But in the same way, we can generate one of these kinds of images from a single script. Later on, I'll talk a little bit about single sourcing and why that's important. Also, what we can do is look at a cloud type image. So, for instance, an Amazon EC2 instance. But the key point is we're doing all of these using a single set of scripts, and we are ensuring that they behave in an equivalent manner. Okay, so some of the tools, very briefly, that we're using to do this are a couple of really nice ones called Packer and Vagrant, which in the DevOps community. The DevOps community are very kind of cool and sexy at the moment. I'm usually a bit skeptical about that kind of thing, but to be fair, they do actually make this process very easy, because I needed to include a little bit of code, piece of code, really all this is doing, I don't want to try and pour over it, all this is doing is formatted JSON, as it happens, is saying in specific situations, so if this is the virtual box deployment, then run this script, run these other scripts, except when you're doing the Docker deployment, the Docker build, I'm sorry, and then run these other scripts. So what we can do is essentially with a single button press, a couple of button presses, we can say, build one environment, and it will make us a image and a Docker container and an Amazon EC2 instance, and it will test to ensure that they're all behaving equivalently, which is pretty nice. The other thing that I would point out here is that there's a lot of support in these tools in particular for other VM engines. So people will often use VMware if they're doing sort of enterprise level or server type deployments. VMware is perhaps more common than VirtualBox. The key principles, I guess, that I want you to think about in this are two things. The first one is this concept of single sourcing. That is, we are using a single set of provisioning scripts, both at the base environment level and at the project-specific level, to build each of these environments to give us confidence that we're going to do the same thing when we press the same buttons. The other one, I guess, is control. Both control in the sense of version control of the environments, and also control in the sense of this hard link between the recipe and the snapshot, maintaining that under perpetuity. Because as you can see, what this gives us the ability to do is go back into our version archive and regenerate any particular result at any particular point. And it's really easy, which is kind of nice. The only other thing I think I wanted to say about this is that I guess the real value here is delivering a result or a software stack in the most appropriate way for any particular user or for any particular result. There might be times when people want your result as a Docker container because that's what that's what they're used to using. There might be times when they use Amazon, or Nectar is an Australian government-funded cloud, which is quite a big thing for Australian researchers. It's great that we can rapidly give people the results, whichever way suits them, rather than saying, oh, you must install this, and bad luck if you're running OS X, and et cetera, et cetera, et cetera. One of the last things I've just about finished that I'd like to draw your attention to is this. Replication and deployment are not the same thing, but they can share tools and approaches. So a lot of what we're doing here, and I'm sure you guys will see this quite readily, has a lot of implications for any kind of automated deployment or validation or repeat checking of things. Because again, we could, for instance, include these scripts in the cat file archive. So you could put them in there, unzip the cat file archive with, in this case, Vagrant installed. You would basically type go, and it would build an environment, provision it, or get the code, run it. And that's quite nice. I like that idea because, again, it's very easy for people to deploy, and also it's controlled. The way that I sum this all up is this phrase, decoupling methods from implementation. So how we choose to do something, our methods, should be as decoupled, as separate as possible from where we're doing it, where in the software sense, in the cloud, in the container, on an OS X machine, on a Windows machine. Of course, sometimes there are always hard dependencies and we can't get around them, but those things are our barriers to reproducibility. And if we can automate and specify the other stuff as much as possible, well, it makes getting over those barriers a little bit easier. And that's all. Thank you for listening. Questions? Good evening, Sarah.
Have you done anywhere, so this is fine for open source and stuff, but
yeah.
Can you do sort of a partial?
Yeah. So a big question here that most people have is what about the various barriers to reproducibility? That is using software which has a commercial license, or using data which has privacy considerations, which of course many of us do against the fungal thing, or using data which is just really flipping big, as in multiple terabytes. You can't have a 3 terabyte VM. No one's going to download that and demonstrate. And so there's a couple of things I'd say. First thing is, yes, they're all problems, but they are barriers to reproducibility itself, not barriers to doing this The second thing is that just because a result is hard to reproduce doesn't make it a bad result, and it doesn't make it a trivial result, not good, but it does make it hard to reproduce. The third thing, though, which I think is important, is that there's always value in including reproduction as part of the result. So either a toy example to demonstrate that your code does vaguely what you plan it does, or you can reproduce the third half of it, or you can reproduce an approximation using an open source alternative. We haven't come across a situation, and we use MATLAB, we use commercial software, we're not a 100% open source shop, and we're not going to. Frequently, we will deal with data that has privacy or accessibility constraints, but we've never encountered a situation yet where there is absolutely no part of a result that can't be valuably reproduced. And that's, so reproducing part of a result is valuable.
So reproducibility is clearly extremely important, no one will disagree with that, but this will allow you to reproduce an error. Right?
It would, yeah.
And quite independent of biological logic, you could have errors, and in other words, it doesn't guarantee verification of the code parts.
No.
It's reproducible, but it's not verified. And that's why the whole standards framework is being developed to provide more control over, or confidence rather, in the codes and the models being able to be verified, that they are what they say they are.
Yep.
Still nothing to do with validation of biology.
No.
So I'm still missing a bit about how this helps in terms of, I can see it's giving you a nice way of encapsulating a useful workflow, but unless that workflow is built on standards and the ability to verify the parts of the workflow, how are we making progress?
Oh, absolutely. Well, I would say that when you do make an error, then having an archival version of the error that you made and having confidence that you have fixed it is really important. So if you build something according to a standard, and by doing so you discover that you have you made some sort of logical error, then being able to go back to the situation where the error did not occur, where the error did occur, sorry, and then verify that it basically broke in this version, it did not break in this version, and we can capture both of those. So I think this might sound a little bit counterintuitive, but I'd argue that the ability to replicate a mistake in perpetuity, once you know that it is a mistake, is itself valid. But this, the point I'd say here is that this is supporting technology to enable, if you like, more validation. It doesn't confer validation in and of itself.
I'm thinking more of verification.
Verification probably is to validation like biology, and that's a very good. Anything else? Yeah, of course.
Yeah, so this is a great talk. I mean, as we've discussed very much on board. In fact, I wanted to throw out the term, we always talk about the notion of software corrosion. And this will solve that very beautifully because you'll be able to capture the entire stack, which is what corrosion is. So, and I really like the recipe versus snapshot thing. The snapshot is what solves the corrosion problem. But to me, the recipe approach is much more interesting and useful. So I really want to replicate it and then give different data, or change the algorithm. So for me, I'm more excited about applications where you demonstrate recipes.
Sure. I mean, I really, the part that I didn't quite get to was that they are they're kind of indivisible, and with sufficient softwaring, as it were, the snapshot is produced as one of the work products of running the recipe. Now, you might not choose to regenerate the snapshot every single time, but if you're doing a particularly important computation, again, going back to a clinical situation, this is not something that is done currently, but I can envisage a situation where, in a clinical genomics context, if you are making, I'm sorry, going to topic slightly, if you're making a treatment decision based on some kind of computation, it would be quite reasonable to me to instantiate a patient-specific virtual machine, do some specific computation, and then archive not only that computation, but also the virtual machine itself as part of the decision-making process for the patient. So yeah, absolutely. The recipe, if it is sufficiently rich, is the place where we start. And one of the other things that we're working on in this kind of area of model combination and validation is being able to, as you say, feed things different data. So a situation where we spin up the VM, or do it on your local machine, pull out some data from somewhere, test the results, and then pull out some different data, iterate through a series of data sets, and determine if we get the same results. I mean, much easier. Now we can deploy that to an EC2 instance or something. If we need 5 extra cores, it's very easy to do it and know that we don't have any link rot or dependency problems and stuff. There was something else I was going to mention that I said to you. I don't remember what it was. I'll come back. Any other questions?
This might be a good way out of scope, but because you're talking about storing things in over a long time period, is it at all applicable to think about storing documentation of the software pieces with the software?
Yep. Well, again, subject to licensing, it would be quite nice to, on the VM, include a copy of the published paper, for instance. Probably for licensing reasons, most journals wouldn't let you do that. But you could certainly, so in this case, that paper is on archive. So we could actually include the archive version of the paper in that digital artifact. Is that the sort of thing you mean? Or?
Not quite. I was more thinking, so if I package up core and my models for someone in 2050 to look at after I'm dead, are they going to know what to do with core that they want to fiddle around with?
You're right, they might or they might not, depending on how important the result is.
That's very
enormous. In which case, the MJ Goulding Foundation, which controls access to all of your digital artifacts, and has an avatar of you, will probably be able to tell them. Facetiousness aside, you're absolutely right, and all we're doing here is giving the ability to replicate things in the future. There's nothing, it doesn't necessarily make it easier for people to do, although the discipline, that's what I was going to say, the discipline of working out what part of your result you can actually replicate with a single button press, which is not everything, and it need not be everything, that's actually quite a good discipline. So if nothing else in 2050, they will get their pseudopods up and they'll say, what happens if we press this button that says run all the experiments? And a whole lot of things will come up and they will see what code generates what.
So the other side of that, if somebody gives me one of these now, I'm reviewing it for a paper, and I run it and it does what they say, I can still be suspicious.
Oh, absolutely.
I go in there and fiddle with it.
Oh, absolutely.
I might not know what to fiddle with.
No, you might not. And again, this is not, none of this obviates the need to produce adequate documentation for your code. But what it does mean is that if you were doing that, then it's much easier for you to go and fiddle with it. You don't have to try and install it on someone else's machine, or and that's really important. That's what I was saying to you. I remember that. The discipline of having to install your code on a really minimal environment that only has the things that you say it needs. That's a really good discipline for software QA, because it turns out that your code needs a lot more things than you thought it did. And we've never done this without discovering at least one dependency that we never realized existed. And those dependencies are the ones which come and kill you with reviews. So it's a hell of a lot easier we found to get our papers through as a result of this. All right, anyone else? Very good. Thank you.
Why did it move? So just before we go online, just a couple of extra things in the program. Yeah. I wanted to mention. So on Wednesday before the cardiac video meeting, we had a bit of a get together with some of the software tools and talking a bit about publishing. So it would probably be worth mentioning as a follow-up to Dan's talk is that Peter Science presented some of the work they were doing in terms of virtual machines and putting sort of the recipe type things in terms of Galaxy workloads on the web as part of their stuff. And I think it works well with the same ideas as what Dan was discussing. Sorry, yeah, and there's slides online, and I will put links somewhere on the website.
Data science actually provided me with one of the most important lessons in my scientific career, which was the first time someone said something snotty about my work on Twitter, which is a very 21st century science thing. And actually, they pointed out that this work was not published in an open access journal.
So we quickly coupled up, I think.
Yeah. To which I had precisely a two-word response: impact and factor.
You talking about the briefings and
Yeah.
because I just downloaded it. So
Yeah, open access.
Around 6 months ago.
Well, I was not behind the paywall and data science. The entire reason we worked out is ironically probably just behind the paywall.
Correct. I'm prepared to believe that maybe the person running their Twitter account.
Now there's entire reason for this one.
So just a couple of other things that came up on Wednesday and have come up a few times recently. And I just thought I'd mention them. Think about them if you want to comment or contribute. Either track us down or post on the mailing list or anything. So some of the features for PMR that are quite often requested is what, you're looking at a model and maybe you spot an error and can you just quickly put a comment somewhere to say that or to say how you think it could be fixed. We do have a tracker, but it's not particularly user-friendly. So some sort of wiki-style discussion feature for exposures might be useful. Or even if you have corrections, Gary's always had quite a few for us. How do you get those back into the repository in the best way? And there are ways to do that, but it's perhaps not so user-friendly again. So if you have any ideas, I'm very open to discussing them.
Like to say something that I really think that yesterday, people want to see it now. You see that as a start.
Yeah.
Should be worth it. I mean, the best way to start, not just people are sitting on the
Yeah.
which is wrong. And the other thing is that there are still needs to understand at least there is
Yeah, and I think now that open core is ready to replace all that functionality, it's probably do a big update.
But really, with the web app, there's kind of web service looking up to the expected results are way too similar to that. And essentially, doing something like the transmap or actual application logs.
So also, I mean, if you're going to use GitHub, there's all sorts of crazy functionality for doing cool things. We don't want to re-implement all of that in PMR2, but are there some minimal features that might be useful that would be not so hard to implement, or doing more to encourage people to actually just put their model on GitHub and they're using PMR2 as a syndication or dissemination channel. And then in terms of peer review, I don't know how many people know it, but we are actually able to provide anonymous access for reviewers. So if you submit any papers to the journal and you put the models in the repository, you can keep them private during the review, and we can give reviewers access, and we can talk to the editors to enable that access. So if you don't know about that and you want to use it, talk to the community itself. And then, of course, it's how do we encourage people to put their models in nice modular, reusable modules that are well curated, validated, mathematically, biologically, physically, adding all this extra information, perhaps adding in data, and also the simulation experiment. And how can we, encourage people to do this? You'll talk a little bit later a bit about some ideas of the young journal that might address this. But again, anyone with any ideas or suggestions, shout them out now or
similar process in manufacturing. This may sound a little frivolous, but we have discussed now the possibility of badges or contribution or alternative ways to reward, in a serious alternative ways to reward sharing and contribution. We're all aware that publication and impact factor is the thing which has to drive us to some degree. But if there are other ways to recognize people's contribution, then that's much more easily done using, I don't want to say the word social networking, but it's much more easily done in digital content. The other thing is, I guess, making the amount of sharing and collaboration to do with the model very visible on the website. So you might reasonably have more interest and more weight by a model if you see that it's being very actively used. And you can see that right away, contributed to by a wide variety of people. If you can see that right away on the website, rather than having to go to the paper, maybe that's a benefit.
I mean, I would also agree with that. So giving examples from our domain, the OpenEHR models repository is a truly domain knowledge governance platform, social media type functionalities that everyone can, even without they can register for free, like Marva. They can adopt, so they can like certain icons, they can receive, choose to receive some fees. But what happens is the platform also supports a truly like version control system, but also a very unique thing that is very much like peer review journal review systems, so that for each model, an editor might pick a certain amount of reviewers from the route and initiate the lead route, and you can have comments to either as a model as a whole or individual model items. And then those are collated and these releases or exposures are released, and those people who have contributed are recognized as a list of contributors, and that's how we encourage people to engage with. But I would say it's all about tooling, and the tool has to be really inclusive and user-friendly and integrated with their way of working social media tools and stuff.
The possibilities to sort of mirror things like CellML, do the interactive issues and stuff on there, and then put it back in as reach some sort of a community center.
Yeah, I mean, it's on a workspace by workspace, it's true, it's synchronized across data providers. But what we're about to level people do that kind of thing.
Yeah, you can script it, the schema you can't push directly to other repositories. So that's one thing to get up there. You can see a way to manage all this relationships. Yeah.
so certainly, now if you want to use GitHub, and some of us do, or some of our just to get better ways to view the version history and stuff. It's easy to do. Okay, so just to start switching everyone's minds over to thinking about annotations and metadata for this afternoon session, I'm just going to quickly highlight probably mostly some of the stuff that Tommy talked about yesterday in terms of the support currently in PMR2 for dealing with metadata annotations. Very good because of that. Yep, so as of now, you are able to add pretty much so if I had more information or more resources in my workspace, they would all show up here. And you can add any of those into this box, which adds it to the RDF index. So it'll basically go through whatever files you give it and look for RDF and stuff those triples into the triple store. Yeah, and you just click a few buttons and things happen. And then that is used as the source data for this simple ontology-based querying, which you can now choose some specific terms and it shows you all the results, as Tommy showed yesterday. Why did the camera just something's going on. We can switch this over. Anyway, so when you do this in PMR2, it's currently using this set of ontologies, where these ontologies have been sucked in their entirety, although I think that was just the self-component part, into this sort of semantic structures in PMR2, and you can do these sort of fixed queries. So there's a predefined Sparkle query that your text is just filling out. But that is accessible via that web interface or through web services. So what happens now when that HAL file you want to suck in is approximately 100 gigabytes, such as Uniprot. And so that sort of approach isn't going to work. As I showed briefly yesterday, maybe this time might be readable. We are starting to annotate our models with Uniprot IDs, things like that. So we want to be able to link those in, and we want to be able to search them, and we want tools like OpenCore that are using the services to pull out the terms to be able to find them. For example, so some way that rather than sucking all these ontologies into PMR2, PMR2 goes out and uses the various sort of tools that are available in the different communities to do the search. We also want to be able to do these user-defined queries. So rather than just this current fixed Sparkle query, we'll hopefully be able to support various other ones and sort of encourage people to contribute new ones. Just need to be a little bit careful because with Sparkle you can do some funky stuff, and we've got to manage the access control and things like that. And then how all of this links in with the SEMGen work, composite annotations, archetypes, and all of that is what we're thinking about. And this afternoon we'll hear about that some more, and we'll work out a plan at the end of that, how that's all going to come together. And to do that, we need to fuel our minds. So now it's on screen.